Hello everybody and welcome to today's webinar, Streamline Atmp Data Analysis Workflows to Save Experiments and Accelerate Commercialization, hosted in partnership with Körber. I am Georgie Makin, the editor, facilitate and moderator for today's discussion. Today I am joined by a great pair of speakers who will shortly be introduced to you. But before we get started, I just wanted to take a second to run through a little bit of housekeeping. First of all, I would just like to remind you that the final portion of this session is reserved for an interactive Q&A discussion, so please do submit your questions via the Q&A widget accessible via the bottom of your screen. We will also attempt to answer your questions offline should we run out of time. This session is being recorded and will be available on demand shortly after the conclusion of the event. Details for on-demand viewing will be sent to you once the recording is available. So without further ado, I would like to introduce you to Christoph, Herwig and Younge. I will now hand over to Younge to start today's session.
Thank you, Georgie, for the introduction, and welcome everyone to our webinar here in regards to talking about streamlining data analysis workflows to save experiments and accelerate commercialization for your industry in the advanced therapy and medicinal products ATMP market. Part of that, what we'll be talking through today is a lot of the challenges throughout this industry within cell and gene and ATMPs, and then how do we actually resolve that to streamline these processes and save costs and accelerate through. Before we start, let's talk a little bit about who we are as Körber Pharma Software and kind of where we're coming from. So we're a company in regards to 700 employees as part of the Körber Pharma Software, but we're also part of the bigger group, which is Körber Foundation. And we have 18 locations worldwide, but our headquarters are in Germany. Our experiences and background is strictly within the pharma biotech, as well as the cell and gene ATMP fields, and we have 30 years of experience with it. And our customer base is global with 120 plus customers, and a lot of them are from the top 30 biotech companies that are out there. And then within our systems, we've done 1,000 installations worldwide as well. A lot of our focus is also on manufacturing execution system, but for today's discussion, we'll be focused on bioprocess data management and analysis. So that's a little bit about us, and let's get straight into the topic of discussion, which is, what are the challenges within cell and gene and ATMP processing today that kind of brings up this forward to discussion. For ATMP, data generation is a very big pain point and challenge that's out there. Experiments are quite high costly, and the complexity to run each experiment, which causes slowness in manufacturing and your journey towards commercialization, is quite difficult. So how do you get the most out of the data coming out is the first challenge there. Second piece, as you run through these experiments, the reason that you're running through them is for process characterization and validation. So a lot of the challenges when it comes to starting material variability, whether it be donor materials, vector materials, or others, as part of the actual experiments, you're trying to take those and you're trying to find how to maintain a consistent quality output of the actual drug product as well. So how do you assess those variations to make the actual decisions that's necessary to move forward? Third challenge is in regards to process variability, whether when you're going through research and development through commercialization, or whether you're using other manufacturers like contract manufacturers or other variabilities or in regards to the corporations that's out there. So how do you assess those as part of the variabilities across the process change, again, in order to maintain consistent control and consistent quality as part of that? And then finally, how do you keep reusing all of that knowledge and all of the data that comes out of it to keep that process knowledge as you go through the product life cycle as well as you scale out. So that's kind of the challenges are that's out there. And what we're going to be talking about is how do you go through these and address these challenges step by step in terms of data collection, information analysis, keeping that knowledge, and then finally maintaining the knowledge management systems themselves and processes. So that's what we'll be talking through and talking about. And I will now pass the presentation over to my colleague here, Christoph Herwig, to continue on. Thank you.
Yeah, thank you very much, Young. Thank you, everybody, for also joining this webinar here. And I also want to just mention that, of course, these challenges are very important, and it needs innovation, right? It needs certain innovative tools along that line here: data, information, knowledge management. I think all of that is known also from regulatory documents like ICH Q12, for example, and also here ICH Q10, of course. And how do we do that? So we will have 4 chapters here which we are following. Let's start with the first one, looking at data. And when we look at data, we want to save experiments because of high hardware and media costs, for example, especially for cell and gene therapy here. And of course, we have the problem that the data generation is outpacing the data analysis. So what we need to go for is that we have to get the analysis results prior to our next experiment. How to do that? So what's the problem? Here, normally the speed of how data is generated is very, let's say, quick, and the speed of data is analyzed is slow. So what is the solution? Here is an automated data collection. Then the next step would be a data contextualization. And then afterwards, the proper integrated, ready-to-use data science, data analysis, which we do in apps. So the first thing which I want to stress here is that we should go for a solution to save unnecessary runs and eventually save money. How do we do that? So we have the problem that we have lots of data sources, multiple heterogeneous file formats, menu interventions, decentralized data storages, and so on. And the data look really scattered. Like this is a picture from a real client here. Lots of Excel sheets, lots of manual operations, etc. And we have not only to look at single unit operations, we have to look at complete process chains, like the process chain here in a cell therapy process, for example. But also we have to look at the dimension of the product lifecycle. We have to link data together from the development as well as from manufacturing. That is the next challenge which we have, so a different dimension. And third, but last but not least, we are not alone here doing that work. We are doing that, of course, with lots of partners. And of course, we have to think about collaborative analytics here. I think that's very important. So how to solve that? We have with Patrick Savvy a system which can automatically import data from all kind of different data sources. I will not mention them here due to time, but we have automatically the possibility to import them. But importing is not enough. We also, of course, have to contextualize. And I will mainly focus on the contextualization and the data analysis. So Savvy is doing the import on the one hand, but is also, let's say, contextualizing the data. So it means what you need to have is, for example, you look at a certain unit operation step here in the development, and you read to relate those kind of data sources which you may have here to a step which is probably at the end of the process chain, and you have to link that to the manufacturing data where we have probably completely different images or data sources. So that way we need first a data model which can actually do that all. Savvy is doing that, so it pushes all the data together in one holistic data model. So Savvy, just to introduce that, is doing the following steps here. So you see here the different data sources, the data collection. It's not a data lake, not at all. It's a data model really on biotech-specific entities, which is based on our domain knowledge in cell and gene therapy here in the biopharmaceuticals. And there in between, of course, we have the contextualization step prior to go to, let's say, different tasks which are probably more in the business focus, like visualization of the data, calculation, analysis, and reporting of the data. And last but not least, sharing the data here within the organization and with external partners. So what Savvy is doing, it is doing all of that together, and that is unique. So we are doing data management, which is more this part, and data analytics in one system. So you may have seen tools which mainly focus on contextualization, and probably other tools which are focusing on extended analysis. But the import in the analysis tool is only as good as the data are contextualized and cleaned, etc. And of course, then you have back and forth imports and exports, etc. And what about your data integrity? So we need to have that in one tool. Okay, so that is, I think, something which is important to introduce yourself. So here comes the first poll for you also. And I wanted to know, how do you contextualize your different data sources along the ATMP product lifecycle? I have shown you one approach. The traditional approach is manual alignment. You, for example, use Excel for that. Or do you have probably already historian data from SCADA or DCS systems and you compare process data trajectories? Or do you follow this heavy way? This heavy way is an automated data import and phase detection of time series, and then the alignment of the features in here with all kind of data sources from electronic notebooks, manufacturing execution systems, or also LIMS systems. Jan, can you just run the poll on that?
Yep. So what folks should see now is the poll is in progress, and basically the votes are coming in. So there we go.
Yeah. So it's interesting to see how you vote here. So yeah, please add your votes. That's great. Yeah, we wait probably a couple of seconds more. Please let us know, how do you manage data? How do you align data here? That would be super, that we get a little bit feedback from you. Good. I think we stop here, Young.
Okay, looks like we got some shared results here, where basically 50% of the folks are basically using manual alignment via Excel, and then 50% of the folks are using the historian data pieces. So no one is actually using the automatic process phase detection right now to kind of move forward.
Yeah, so this is of course a question which is somewhere in between the data management, and the next part which I'm going to show is of course the data contextualization, and this one of course concerns the data series contextualization, etc. So I will continue here and have a look what we mean with this. So data management is very important, but then the question is, what do you make out of the data? And therefore, I jump into the next section, which is the section on information. So you have the data now in. How do you really now contextualize the data and look at, for example, variations in a certain process step and make sound science decisions? And I took here an example, which is the starting material variability in cell and gene therapy. Your blood cells are different from mine. I think that's clear. The cellular attributes are very different. And we still have to set up a proper process. So you see here the classical targets in ATMP process development. We have to quantify cell growth. I think all of you are concerned with these points: shorten process duration, assess process dynamics, optimize media, optimize productivity, optimize product quality, and so on and so on. But what we have underlying of that, a lot of data here, as we see, and we have to make proper decisions out of this data set here to get to these targets here. So what do we need to do is contextualizing the data. And I will show you our approach here, which we follow, probably uniquely follow, as 5 steps in data contextualization. So the first step is an automated phase detection from your data sources, from your time series, and then you can detect, for example, a batch phase or a fed batch phase or induction phase, transfection phase, etc., decay phase, etc. This one can be put of course, manually, but we don't want to have a manual thing here. We cannot keep up with the speed of generation. So we have to put probably more smart solutions in there. We have different technologies. We can have importing events. We can use dynamic threshold models, fuzzy rules, kinetic modeling, for example, in order to identify the different phases, etc. So number one, you have to set the phases. Second step would be to calculate your KPIs of interest here inside of these phases. So here, for example, in the upstream part, that's a classical AMBER high throughput bioreactor example here. You have to transform your viable cell count concentration here to a specific growth rate, for example, and you have to calculate, for example, derivatives, etc. There are different methods also out there. I will not go too much into detail, but we have to calculate these kind of KPIs from here. There are tools inside of Savvy directly available to convert that here automatically. We are also using for those kind of elements, because not always we can use mechanistic tools here, like of course mass balances, etc. We are also using here artificial neural networks. People would say, oh, we are using ML or AI now today for that. But this is a very simple artificial neural network where we can properly estimate rates from the data, where we only have time as an input here, and then you see that we properly could here estimate, for example, for a cell culture run, the different rates. So that would be the second step which we are doing. The third step, what we can do is, of course, looking at one phase particularly, and as you imagine, we need now to do a very smart step, which is unfolding of dimensionality. As long as we have the time inside of this kind of data source, we are not really heading for a proper solution. We have to get to features, and the features are here. So we unfold the dimensionality. That means we are not having time anymore, but we extract certain features, a minimum, maximum, average value in a certain phase of our KPI. Here, for example, specific growth rate. This one can also be done automatically. And this one you do for, let's say, one reactor. You do that for one KPI, but probably also for your different CQAs. And then you append this kind of list of features with all the elements what you have changed or what you want to look at it in order to make sound science decisions, like clone, media, day of the week, whatever is here important is metadata. And then once you have that in this kind of feature metrics, then you can start working with it. Before you do that, of course, you need to do the fifth step, and which is the case not only doing that for one reactor, but doing that for all of them. But as I mentioned, this is all done in an automated way, no manual interaction here in there, and then you are ready to go and you have a feature matrix which you can use. Like this, you can speed up your data analysis and keep up with the data generation. Of course, then after the feature metrics, you can of course use Savvy also for analysis of the data. Here, for example, you can look at, of course, the outliers of a certain KPI here over time and compare that in a golden batch form, if you like. That's one way. But of course, to make a proper decision in process development here, for example, for clone selection, you can group the different results which you have here, the features, and make proper decisions saying, okay, this clone here, this clone number one, seems to be having a significant higher specific growth rate here. So I may select this clone for that purpose. And this one you can do automatically. So that is one example on contextualization, which I want to bring you. And that is about the information on of what we can retrieve from the data. But information is only halfway of what we really need to demonstrate also to the regulatory bodies, where we actually really have to demonstrate that we have our process, our complete process under control. That means we have to look at the production of a robust control strategy. And this one, of course, has to address, let's say, not only individual unit operations, but it also has to address the complete process chain, so all unit operations. And of course, it should include variability across the process chain. So let's have a look what we can do with that. Once the data are managed, we have contextualized them, and now what kind of knowledge we can really retrieve from there in order to get to a robust control strategy. The task is probably also shown here on that slide, especially focusing on cell and gene therapy here, where we have the variability of your starting material. Then we have probably also the subsequent, let's say, processing step of expansion and formulation. And what do we have to do is actually to run those processes in such a way to compensate for what is really happening here, right? So we have to have a solution to get finally here to a consistent quality product here, which is robust. Okay, so therefore we have to have process understanding, and this one can be shown, for example, in classical Pearson correlation matrices, also in Savvy here in a univariate way. You can have a look at a certain acceptance criteria, let's say distribution of your CQAs and your intermediate CQAs here across unit operations. So these ones would be the different unit operations. On the x-axis, it's actually a coordinate plot, a parallel coordinate plot here, which we can do. We can also formulate a control strategy here using our Sound Science control strategy app, where actually have here the different CPPs, and the green phase is a green part is showing you the PAR with proper tolerance intervals considered, and for a certain CQA which we should use. Okay, these are tools which we have available already in order to ensure consistent quality. Of course, this one can be formulated as such, and if you look at PQLI AMP study 12 years ago, I think this plot is already in there, but I think this is also still valuable. But we have to go beyond of this, and we have to show sound science distributions and proper PAR estimations here. But I want to go even beyond of that and have a bigger look at the control strategy here in this chapter. And I want to show you what is the bigger task here. The bigger task is to have process understanding and control strategy set up. And what do we need to do here? We have to link CMAs, CPPs to CQAs. So it's nothing else than probably this plot, or let's say this plot, but we have to show that not only for one unit operation, but we have to show that for each unit operation and for the interactivity between the unit operations. So how can we do that? For single unit operations, we can take the former example back here, and it's the Atmp study for the upstream, for example. And we can just append our metrics, which we had before with CPPs. We extract also features from these here. We append these CPPs also in this matrix, and then we identify CPP, CQA relationships, what I have just shown you, with these kind of plots, and you use principal component analysis and MLRs, like you may know them. I think that's for single operations. But what do we do for the complete process chain? So what's the problem? When we look at the complete process chain, I made it a little bit easier here to show you that one with a classical process chain with certain parameters here. For each step, you may have 10 process parameters here and here and here. So maybe it's probably 200 process parameters, which you have to keep in a range in order to make sure that your CQA is properly in line. And of course, this is what I show you here should not happen. And this one happened in case you are changing these process parameters here a little bit, and then they go off. Okay, so your control strategy should be stable enough to be able to compensate for the variations which you may have inside of the variations inside of your parts, of course, which you have probably defined for your single unit operations. So this is something which we have also in Savvy. We have a possibility to depict complete process flows. Here, the upstream part, a filtration step, purification step, for example, and then you can analyze all the steps here together and do a complete process control strategies here across the unit operations. I will show you one very detailed tool here, which we really think this has lots of potential, and that is the integrated process modeling. And the integrated process modeling is based on individual operations, which we actually analyze on CPP-CQA relationships. And then we run Monte Carlo simulations over the entirety of the process here. And like this, we can, for example, analyze what is the impact of a change of a CQA of that step here on the final drug substance CQA, for example. So if you change here, for example, the mean of that step, what will actually happen with the certain drug substance quality here at the end? And from there, you can deduct for that step a certain acceptance criterion here already. So that's one thing what you can do with an integrated process model. The second item, or let's say what is linked to that, is as a result from this, you get here, as I just mentioned already before, you get from this parallel coordinate plot, you get for the certain CQA, you get certain intervals out for each unit operation. And these are the intermediate acceptance criteria which you may have here. And I think this is very, very, very useful. And I really wonder who is doing that here on a model-based way. Okay, normally you may have put 3 standard deviations on the mean, and then you say that's my intermediate acceptance criteria. But we have found out with the model that you can sometimes relax this kind of specification, and sometimes you need to constrain this this acceptance criteria even more. So be careful when you use classical, let's say 3 standard deviation approaches only. Okay, so that is one thing what we can do here for intermediate acceptance criteria along the process chain. We can also use this integrated process model where we have the same sketch here. And now we are not changing the CQA distribution, but we are changing a certain process parameter step here, for example, at that step. Okay, we change a process parameter, for example, pH, chromatography, whatever step here, we change here. And then we look what is the impact of this CPP on the final drug substance. And there we also see that also with this approach, we can finally say, come on, the drug substance quality has a certain out-of-spec probability. We can say, we only want a very small part to be outside of this. We want to have Six Sigma approach, for example, and then from here we can deduct what proven acceptable range we may set for a previous unit operation here. So this IPM is a real useful tool, number one, for intermediate acceptance criteria, and here also setting your control strategy. Good. I think that is what I can show you for that. And of course, the next step after having a control strategy really set up properly is, of course, doing also a demonstration that your process is properly in place. So that would be probably a later stage in manufacturing, for example, continued process verification. And there you, of course, want to trend certain variables, CQAs, and over these different over the batches, and you want to show, for example, the Nelson rules that you have your process under control. That is fine. But this just simplified, let's say, univariate approach is, in my opinion, not really the way to go. We should have one step more. And we developed a unique tool based on principal component analysis to look at the entirety of the data and to look at, let's say, outliers which may come from there, like this here, and you compare not univariate values here, but you compare the entire batch to a control group like this one here. The green area would be, for example, your PPQ runs, for example, and then you compare certain batches and you find out, this has an outlier. And then you, of course, want to know where would this outlier come from? And the app here in Savvy directly helps you in that to identify the root cause by just clicking on this, and then you will see what are the main contributors here. I mentioned that this app here is based on principal component analysis, a very easy tool. However, we are using here a more advanced tool. It's a robust PCA, which helped us also in case studies with the industrial partners to really find out proper outliers, which were not really found before. So what this chapter is telling you is with this approach, so the integrated process modeling, which I have shown you before, so you have the different operations, you have the process parameters, etc., and also with this kind of CPV app, you have your process under control. You can ensure consistent quality. Here, with this tool, setting up the control strategy properly, also by looking at the interactivity between the operations. And with this tool, you can really find out early enough certain root causes to make sure you are not failing with your batch. With this one, I also want to run the second poll here on that, how you actually set up a control strategy. And what do you do here? Probably you can help me in answering these questions. So number one, you first only compare single operations. That would be one way to do. The second item would be that you actually run a multitude of linkage studies. Let's say you run experiments, a lot of experiments with all kinds of variations between the operations, and you look how stable your control strategy is. Or third, do you use the shown integrated modeling approach to focus on holistically derived critical process parameters and limit the number of experiments? So you have here much less experiments than here. Please let me know what is the current approach you're running here. So there are some answers here on the linkage study. Yeah, this is always a question, how many runs you really want to do. I think that's very expensive, of course, right? So I think you should try the integrated process modeling approach here, probably even more. Let's wait a minute more to have a look at your responses. I think there's, can I encourage you to vote? Else, Young, I think we are just stopping here.
Yep. Give it a few more seconds and then we'll go. Okay. So if we stop the polling right now, I'll share the results. Very similar to the last time, exactly a 50-50 split as well, in terms of comparing the single unit operations and the second item where you're linking it across the process for control strategy.
Yeah. So you should try out this integrated process modeling approach. It's really helping. It is using your knowledge from single operations. It links it together via Monte Carlo simulations. And like this, you save experiment and you jump over that step and you only need some verifications here. I think this is already a big step to save costs and also to accelerate commercialization, which is finally the goal of this webinar. Okay, thanks, Younge, for that. I will go to the last part, which is the knowledge management. So the control strategy is your knowledge, right? The CPP-CQA relationships is your knowledge. How to capture that properly and provide that also to have synergies with other products or other processes, etc., in a kind of scale-out concept here. Let me have a short, simple look first on that. What, of course, can be done, and this one would be, of course, analyzing two different products or sites or scales, or and analyzing just a statistical on a statistical way two performances. Here, for example, with a TOS test, so a 2 one-sided t-test, which actually checks significance on equivalence, right? And here you see, okay, here this site B is inside of the tolerance of the site A, for example. So this one would say, come on, there's a statistically similarity given. That is one way, and of course we have these tools in Savvy as well. But that's only, let's say, one step to do and to explain what you can do by checking out if your your tech transfer, your scale out, etc., would be okay. I would like to show you a different approach here, which is more on the knowledge management based. And there I come back to a publication which we have done here. It looks a bit complicated here, but we published this one in Pharmaceutical Research, and it actually had a look on what kind of tools are available in knowledge management here, and how this may help you, how these tools may help you to for example, here help you setting up a control strategy. And there are a couple of tools out here. I will not mention all of them, but you see there are statistical analyzers which are used, and intelligent agents, like, for example, also ontologies here, which can be used. And there are, of course, also here models and simulations. And we are using mainly for knowledge capture on CPP, CQAs, we are mainly using models, or let's say today we would say digital twins. And you can see that actually they are helping a lot. Finally, here when you go around, they are finally used very much here, innovation support. So this one helps in the scale out concept in this sense. Okay. So how do we do that? We actually developed these kind of modeling approaches, which are needed here for that approach. We want to set up a model, so we know how to set up these models. We have tools and workflows along GMP, good modeling practice approaches, not GMP in the sense what you thought. So it's a good modeling practice. We can set up these models, and with these models, we can capture the knowledge. And the second item is we can deploy the knowledge accordingly with these kind of models. We can also append those kind of models with hybrid approaches. So we use artificial neural networks, we use machine learning, etc. in there, in order to actually adapt the models also from, let's say, one product, and to run them also on the second product, and allow scale down, scale out techniques. Okay, so there are tools here now around with hybrid models which we use to scale out and deploy the knowledge which we gained here. These tools are available in Savvy. You can run all these models in the Python editor, which we have. They can deploy it as apps to everybody who is just using them. So we don't need everybody who's just coding, have a coding experience. And then you can run here, for example, those models as digital twins, and you can have predictions, and you can run those integrated process models also here for predicting what is actually happening even in real time. So these are the tools which I want to show you here, also for knowledge management. Use digital twins, further on also ontologies, which we are going into, and then you deploy them for a different task. You adapt the models, the digital twins, and then you predict what will happen even in real time. So this is a kind of summary of that. So we capture the knowledge in the models, we adapt the models here, for example, with machine learning to a new product or process task, and then we deploy them to predict or control the process. So this one will finally help you, of course, accelerating commercialization. Coming to the conclusions of this talk. Number one, I think you have seen that we actually have a tool with Parsec Savvy, which does data management and data analytics together. Yeah, you will have a big gain in saving time in data management, data cleaning and preparation. You will have standardization of workflows across all your team for visualization and analysis. Because you are not going back and forth in different tools, we are not having a problem of data integrity, which is very important. And of course, this one helps accelerating commercialization as such. We position Savvy in the world of digitalization here. You see the blue parts. This one would be the Savvy part. So we are linking to different data sources. They can come from data lakes, historians, data warehouses, whatever you have available. They can also be directly coming here, say, from different data sources directly. So we are not really depending on this data layer. We can also go around of this. And then what we do is this domain knowledge-based data management, which I have shown you with the contextualization and alignment. Then we actually do the knowledge step, the information and knowledge step, which is the data intelligence. We do data analysis, dashboarding, visualization, sharing, and reporting. And finally, we of course head for your business goals, which is of course out of spec handling, control strategy setup, quick life cycling, continued process verification, process control, etc. All these elements are more on your business side, and we help you with Savvy by deploying you all of that, no matter what kind of data sources you have. We are innovating with partners. I think that's important. You have seen here innovative tools, which we develop here with a PDA, for example, with ISPE. We have done that. This is a root cause analysis publication, which we did with Takeda here, for example. And that is the integrated process modeling here also, which we did with Boehringer Ingelheim. Have a look in, just contact us. But you see, we want to be in forefront of innovation here to help Atmp processes to be a success. In this way, we are also innovating here inside of different organizations. I mentioned already ISPE. You see here also the ICT, the NIMBLE, where we are active, the Alliance for Regenerative Medicine, and here the Manufacturing USA, where we are proud members of. Finally, I come back to the frame of this talk, probably the last slide on this. The data, you have to have an automated and combined data management analytics tool. And then you have a holistic data model for Atmp processes. This is what we can provide at the beginning. Then we can provide you with the domain-oriented data contextualization workflows, and they help you to directly make sound business decisions, like I have shown for example, a clone selection. Then you have to have a look in the knowledge part. How do we really set up a robust control strategy? We are using innovative multivariate statistics, robust PCA, for example, and integrated process models, for example, for that. And last but not least, we want to have event prediction and platform knowledge. And this one we do here with the deployment of digital twins as knowledge management tools. Thank you very much for your interest in our work, and hope that we can discuss in the Q&A right after. Thank you very much.
Thank you both for your presentation. I think that was a great introduction to the second part of today's webinar, the discussion segment. So before we do get started, I can see we already have a few questions lined up, but I'd just like to remind the audience that you can still submit your questions to our panelists via the Q&A widget at the bottom of your screen. So the first question that we have in asks for, well, what is an approximate return on investment that can be shared, or alternatively, do you have any examples based off past projects?
That's a great question, Georgie, so thank you for that. A lot of the items here in regards to return on investment starts with benefits, right? Where are we getting the benefits out of this? And we talked about saving experimental costs as well as acceleration through commercialization. So from that perspective, that is kind of the focus areas. The other things, if we're talking about the return on investments, it also depends on what you're investing in. So as part of the PASIC Savvy implementation, if we're doing a subscription model, or if it's, for example, hosted on the cloud versus a holistic implementation and you're hosting on premise, the return on investment is quite different there. Typically, we would see the return on investment more or less to 1 to 3 months in that sense, once you actually get everything going. And that's kind of the items that we want to see there. Yeah, Christoph, is there anything that you want to add in terms of more details in regards to the?
I think when we are already having, when we talk about experiences with our project, which we are doing, of course, there is always an implementation project to be done, right? The implementation project and the complexity of the implementation project is based on the number of data sources which is normally to be connected. That's number one. Secondly, we have already a lot of data sources connected. So let's say the synergies are there and we don't have to reinvent the wheel. We can directly start from there. So that means this kind of implementation project is very very short and neat, and should not hold us up here. The implementation project is the one which is, of course, in these 3 months return of investment, I think, as you just mentioned, because the subscription itself, when you think about the subscription itself, the cost for that are only a very minor part of the hours you save with the tool directly. That means if, for example, just make an example here, you save per person per month, you save 20 hours with using Savvy per person, and the cost is only a very minor part of that, probably 10% of that. So the return on investment is coming from the first day.
So as a follow-up to that question there, you've just directly mentioned saving time and cost associated with time, but where are the other opportunities for saving costs?
So the other opportunities in regards to saving costs is specifically regarding the saving of the experiments, kind of what we talked about from Christoph. When you are taking a look at the experiments that you need to do, right? With the current model, what you have to do is take a look at the experiments, actually run them physically as part of it. But what we've shown as part of the presentation is with the integrated process modeling and the digital twin technology, we're able to virtually kind of analyze those experiments and provide some predictive analysis as that part of as part of the tool set. So with this, you're virtually running those experiments so that you can then take those analysis and focus it on target experiments to save those costs as well.
Excellent, thank you. And circulating back to you, Christoph, you mentioned about system implementation. What would be a typical timeline and I suppose level of effort required for implementing that system?
Yeah, let's say depends on the complexity of data sources. I think that's clear. But normally we have that up and running in 4 to 5 months. So we normally involve us very much in the user requirements. So really finding out what are the core plots which are really needed. So we are also knowing what is going into IND, BLA filing, what are the plots which are really required for demonstrating process understanding or control strategy. And from there, we actually take our partners, the clients, to say, come on, how do we streamline the implementation in this direction in these 4 or 5 months, which we have together. But let's say normally we take these 4 or 5 months to our side. So the time our clients have to spend for this implementation are typically only a couple of hours after this joint user requirement phase, and then we will do the rest here mainly. Okay, and probably probably just also to come back to the part was Younge was saying that of course we are saving experiments and we only verify these experiments once we have, let's say, a good idea from the digital twin or the integrated process model. We are setting up a system which is having very robust outputs, and we are doing that very quickly. So I see also with our clients that there are hardly any tech transfer surprises. That means the control strategy does not need to be readjusted. And that means that we actually really accelerate commercialization along the product lifecycle, along that line, because you have really short phases and you don't have to, let's say, reiterate on the control strategy again, which is normally the case. If you're only focusing on single operations, you will finally have in the manufacturing scale, when you have manual interactions, if you have probably different equipment, etc., you will have the interactivity between the operations, which will finally lead to a failure of your batch, although, let's say, the control strategy for a single operation was okay and held properly. So these kind of surprises we don't have with this approach.
Wonderful, thank you. At what stage in the commercialization journey would you anticipate data streamlining becoming a priority?
Really the very first stage as part of research and development, right? We mentioned kind of the topics of process characterization that's related to ICH 12 guidelines that's submitted by regulatory. When you do process characterization, that starts at the research development phase, going into phase 1 of clinical trials as well. So getting all of that data, getting all of that analysis, predicting and doing that types of research at that point in time and analysis at that point in time is quite critical. And the earlier that you start with this, the more that you can get out of it, the more data that you can get out of it, which means the more analysis that you can get out of it. And that's why you can use this tool to streamline and accelerate towards commercialization.
I can probably just add on that. We have partners, they invested millions in the equipment, in PD, right? They have invested millions in automated analysis, automated high throughput bioreactor systems, in screening tools for chromatography, purification, etc. Those systems and the hardware is there, okay? And I've seen many times these things running, but the output is not used. So coming back to the point that actually we have a high number of data which are generated, but they are not exploited, not analyzed properly. So that's why Younge is completely right. There we have to start in order to use our resources properly, okay? And then secondly, of course, it's important to set up a proper control strategy in stage 1 validation. And that starts first with pure development data, and then slowly manufacturing data will come in, and then you tie them together. And so in this way, you have to have a platform available where you are able to tie that together. And the data sources from development and manufacturing may be different. And you still need to look at that together. And you need to contextualize this together. And if you just do it when you're already manufacturing, that's much too late, and your control strategy will fail.
Excellent, thank you. Sort of leaning on the answers you gave there a little bit. You've mentioned, we've got it on the screen there now, actually, data, information, knowledge, and knowledge management as key focus areas. But which would you say is the most important there?
Yeah, go ahead.
Yeah, so if we're talking about, let's say, the typical market for cell and gene and ATMP, right? If they're starting out in that sense and they don't have infrastructure, then the first starting point would be, hey, they actually need to build the actual infrastructure, get these equipments going to actually start collecting the data. And then after that, you start getting it into data contextualization so that you can use that data in that sense. And I think this is where Christoph is going to segue into about if you're talking about, hey, you already have all of that data, right? Then that's a different answer in terms of the next step in regards to data contextualization.
So probably if I would say knowledge is the enabler for getting your process validated. Before, you cannot do anything, okay? And knowledge management is your enabler to have the right business. So you say, come on, you have the synergies between the processes, okay? So, and the other ones are, of course, prerequisites to get there, okay? So knowledge is the one which you need to demonstrate that you validated, and then knowledge management is your business case to other products. So I think that is how we can split that.
Thank you. It's a very comprehensive answer. You mentioned that the outcome relies on data models or process models. If you don't have a model, where do you start?
We have always models. Let's say they are just of different complexity, okay? So we can always start with some data-driven correlations. We can always have that, okay? But of course, we have the problem that we have very, very many parameters. I have shown 200 parameters and only a very few observations. So these cultures take a lot of time, right? And so on. That means we have the classical N2P problems, or less observations and many parameters. That means just a classical, let's say, data-driven Google type machine learning will not help here in cell and gene therapy, not at all. That means we have to constrain our problem. That means the model which we build has to have some domain knowledge in there, right? And I believe our partners have this domain knowledge. And then we put this domain knowledge into the workflows. We have these good modeling practice workflows around there, which include data-driven and mechanistic part in order to develop those kind of models. So we always have a possibility to get to a model. The main point is where we actually spend the most of the time with our clients is the proper goal of the model. So what should the model really show? And normally that is, of course, a kind of CQA-CPP relationship or CMA-CQA relationship, which we really want to have a grip on. And this is a direct, let's say, response or an input into the control strategy. But normally you should not be really afraid to say, oh, I need to set up a model, I don't know how to start. This is solved. I can tell you this one, we have solved that issue.
Excellent, thank you. I do have one final question just before we wrap up. If you are starting from a position of not having a sophisticated model, does implementation time change, or does the process change in that respect there?
Of course, as I mentioned, the model is a good tool, but we can always have a data-driven approach first and then refine it. It should never hold you up. We have to understand, when we think about ICHQ12, also it's a lifecycle management approach. If we think about FDA discussions, which we did around QBD, quality by design, it's never one way. It's always an iterative approach which we do. So the model first is simple and then it refines over the life cycle. And so therefore, I don't see that this is really a showstopper here if we don't have an appropriate model right from the start.
Excellent, thank you. Well, unfortunately, that's all we have time for today for this session. Before we wrap up, I would just like to take a second to thank our speakers for their insightful presentation and answers to our questions. As always, you can access this webinar on demand via the Facilitate website, along with many more of our previous webinars. Until next time, thank you for joining us and goodbye.
Thank you all.
Thank you all. See you.